So my name is Alex Frangi, together with Mark Horner, coordinating the whole event. So both of us are both members of Avicenna Alliance and the UK CEiRSI, or Centre of Excellence on In-Silico Regulatory Science and Innovation. But for the purpose of this event, Mark is representing Avicenna and I'm representing UK CEiRSI. So I'm not going to go into the details of the agenda because you should have it. It's in the invitation. But each of these sessions will be provided by a group of colleagues that are members of Avicenna or CEiRSI or both. And we also will have at the end 15 minutes for an open discussions and questions. The history of this event, just for those of you who are not familiar with it, is that Avicenna Alliance and UK CEiRSI together, and just to be more specific, we are a consortium of public and private organizations. UK CEiRSI has more an ambit of action which is within the UK, but we collaborate very closely with the Avicenna Alliance and Avicenna Alliance more internationally. We work with actors both from the private and public sector of various companies, both on the device, pharmaceutical, but also on the modeling and simulation community, alongside academia, and in particular with the VAPH, or the Virtual Physiological Human Society in Europe, and CEiRSI as well, to develop regulatory science and to provide support to colleagues in industry and regulators for the adoption of these techniques. So we put together last year in Japan a presentation to IMDRF, and there was an agreement with all the member organizations of IMDRF of running an event for training that would be targeting, and particularly colleagues like yourselves that are all in the front line of reviewing or assessing sort of dossiers for medical products. Today, we're going to focus primarily on medical devices, but we are planning to do similar sessions for pharma in the future. You will see that the examples may change, but the principles are very much the same. Therefore, if you are from the pharmaceutical domain, I think you still will learn a lot from today. We are not assuming any prior knowledge, but we are welcoming questions of any type. Now, we also want to emphasize this is a limited event in the sense we have 90 minutes, and you will see that it is quite action-packed. But we are planning for a two-day event in London in October that will be more hands-on. And if we find that is successful, as we hope it will be, our plan is to then bring that two-day event to other geographies to make it more accessible as well to people that may not have the chance to travel to London in October. But it will be open in principle for everyone, not just from people in the UK. Okay, so without further ado, I'll hand over to my colleagues who are going to be introducing you to the topic of in-silico methods.
So what do we mean by in-silico evidence? Well, we're going to start off with asking what is a model, and the upcoming definitions we'll talk about are computational modeling and simulation, the difference between a mechanistic model and a phenomenological model. And two of the concerns for regulators we want to highlight is, how do I know a model reflects reality? And, well, no prediction is correct, so how wrong could this model be? So computational modeling and simulation, basically a numerical representation of a real-world system in a computer. Two types of models then. Mechanistic models are based on cause and effect, so physics and first principles, and phenomenological models are predictors that don't necessarily make any causal assumptions at all, so statistical based predictions. A classic mechanistic model would be numerical weather prediction. So we divide up the atmosphere, sea, land as well, into a grid, and then we try to predict the weather. So how do we know a model reflects reality? Well, in this case, hardwired laws of physics, first principles, maybe Stokes equations. That seems like a pro. Maybe a con to this is actually you can't model all processes or all timescales and space scales. So it can't be, if you like, fully representative of reality. No prediction is exact. How wrong could this be? Well, a pro is that we normally forecast chances of weather events, and we can check if those forecasts, those confidences were correct. Con, there's a lot of ways you can represent that uncertainty, and a lot of ways you can get it wrong. Similarly, Earth systems model, climate models, similar on the how do I know a model reflects reality? You know, it's based on laws of physics. Con, we can't model everything. How exact can, how wrong could this be? Well, we do check how wrong these models are with what's called hindcasting, checking they can reproduce past known temperature records. But a con, well, we can only really validate our confidence intervals in hindsight, and the future hasn't arrived yet. Hard to predict the future. Now, a phenomenological model, like a statistical model, might be something like red sky at night, shepherd's delight, or I've heard it's also red sky at night, sailor's delight. So this is trying to predict the weather with a rule of thumb. How do I know the model reflects reality? Well, in a purely predictive model, maybe something like AI, or a rule of thumb like this, you only care about correlations and not about causes. But on the other hand, as a pro, AI weather prediction is actually getting more accurate than physics-based weather prediction, at least for like regular weather. But no prediction's exact. How wrong could it be? If you're using a model that's only been trained on historical correlations, it might not be able to get things right. So it might not understand red sky at night, barn on fire as an alternative explanation. It just looks at, well, there's a red sky tonight, so it's going to be good weather tomorrow. But there might be another reason there's a red sky tonight. And as a last example about forecasting and prediction, this is called Penrith weather forecasting stone. So it takes in a condition like stone is wet and predicts rain. Can't see stone, foggy, stone is gone, tornado. So does it reflect reality? Well, it does seem to be just a sensor rather than a real predictor, but it does at least explainably link cause and effect. So that's good. Explainability is good. It only really achieves 100% accuracy by predicting the past or the present, right? So it's just a reminder that, well, how wrong could it be? It's important to realize nothing will ever be 100% accurate if you're trying to predict the future. But at least its predictions are constrained to something plausible. It only predicts weather we've seen before. And it is a caveat here. This isn't just a sensor. This is a predictor because it basically takes something we observe, which is an effect, and just predicts the most probable cause. But, you know, stone is gone. Is it a tornado or did someone just steal the stone? So it's only predicting that there was a tornado. And the modern view really is when these models are applied in medicine, a lot of models will sit somewhere between a mechanistic model and a phenomenological model. It won't be necessarily one or the other. So sometimes we call a mechanistic model a white box model, highly transparent and interpretable in theory, and a black box statistical model would be at the phenomenological end, hard to understand what's going on. So where does it fit in? Upcoming definitions, in-silico and digital evidence, also called in-silico evidence. And the concerns we want to highlight here is any of this proven in practice? And is the model credible for the specific decision in front of me? So in silico means in a computer, in a way, in silicon, instead of in vitro, ex vivo, or in vivo. And digital evidence or in silico evidence is outputs from your simulation that are included in a regulatory submission. So in practice, something that's quite familiar, I've called it in-tunnel versus in-silico. So wind tunnels, we can put vehicles in a wind tunnel, and we do and have. We can observe aerodynamics, but we can also do that in a computational fluid dynamics simulation. We might get a lot more information out of that. It's easy to see how these are comparable types of evidence. But why not also apply this to medical devices? So you could have a stent in an artery, for example. In this case, we've got a transcatheter aortic valve implantation instead of a car. And instead of looking at aerodynamics, we're looking at hemodynamics through the aorta. Broadly similar. And another example, crash tests. We've got cadavers versus simulations. And I did pick cadavers because I think it tells you an important lesson. I didn't realize that originally cadavers were used instead of dummies for crash tests of cars. And one of the reasons we moved away from cadavers is not just the maybe ethical implications of it, but because they just weren't representative of the population of crash test victims. They tended to be older people who died naturally, quite frail. So dummies allowed you to be more representative, although even then they tended to represent typical adult males. But we can also do crash test simulations instead, or as well, I should say, and get more information this way. And you could think about this as how it links to a virtual cohort. It's not always going to be possible to get a clinical cohort that's necessarily representative of edge cases, rare cases. And you might want to supplement your real patients with a virtual cohort so you can actually explore these rarer cases. Complementary evidence at the end of the day, bench experiments, animal models, clinical trials, there are all types of models used to generate evidence. And the question really is, for computational models, is this computational model credible for the decision in front of me, just like these other models have to be credible?
What drives trust in a model? We'll talk about some important definitions that are relevant to the way we discuss trust or credibility in the model. Things like question of interest and context of use, verification, validation, applicability. We'll talk about some concerns that we want to highlight. So many concerns around modeling and simulation relate to the idea that it looks like a black box. So how can we trust the way the model is constructed, the way we interpret a model? So to get to that point, what we want to do first is to go into a framework here where we think about decision making and the way in which assessors and others think about a particular decision and the relationship between what's observed in reality and a decision that's being made. So importantly, on the left-hand side here, in this very simple representation, we note that regulators and others do not assess decisions directly from observations. They don't go straight from a direct observation into a decision about the release of a product directly. Instead, what they do is they make inferences, right? So all of you, as you're thinking about measurements, as you're thinking about the assessment of data that comes in through a submission, you're going to think about the data, but you're going to think about it through a lens. And that lens is going to be the inference you make off of that data that helps you to arrive at a decision. So the key point here, we're not making the decision directly from an observation or a measurement. But when we think about decisions, there's two key terms that our community in the modeling and simulation space use in a very specific way. This will come up later. We'll see it repeatedly throughout today's discussion, throughout any of the work that's related to modeling and simulation. First one is a question of interest, relatively common term. As we think about it in the community, it's very much a broad question or decision or concern that's being addressed through the use of the model, but maybe through the use of other things. So it's the very broad question you're trying to answer. A more specific or refined aspect of that is the context of use. And now we're starting to pull in the specific aspects of the model where you're saying, how does the role and scope of the model, computational model or otherwise, how is it used to address the question of interest? So the context of use is the specific application of the model to the question of interest. Two key terms there, we'll come back to them. Now, what's important to think about here, and the way, one way to look at this at the very least, is to say, all right, so we have our measured components as we saw previously. We have our decision we want to arrive at. Importantly, when we think about a model and when we think about what goes into a model, that model is actually going to knit together a number of important things. not only is it going to knit together the measurements, so what we're observing, it's also going to pull together what we don't know and things we might be able to say about the scope of what we don't know, so our knowledge gap. It's also going to pull together on the right-hand side here the chosen aspects, the things that we made decisions on in building the model. So the model, that triangle in the middle, is going to pull all that together. It's going to present somebody who's observing this with an inference, or the information that allows them, rather, to make an inference, so that ultimately they get to the point where they're able to assess a decision by inferences that are derived from the observations through the use of a model. So we want to think about this big picture as a model is taking a measurement, but it's also pulling together these two other aspects of the things that you've chosen to put into the model, and also needing to accommodate the things that you don't know, or your knowledge gap. So let's see this in practice with a clinical study. So when we think about a clinical study, a typical traditional clinical study, randomized controlled trial, there are a number of things that you've chosen in, or the person or people running the study rather, have chosen in creating that study, right? There's inclusion-exclusion criteria. There's what you might choose to use as a control group. There's decisions around endpoints. The decision-making process is robust for what you choose to put into a model. The things that you might measure are things like adverse events, functional scores, et cetera. And ultimately, there's a number of things that you just don't measure or you can't choose in the model. Things like the way the patient's going to behave, things like their underlying physiology. Now, when you make a decision on the basis of all the information that you have from a clinical study, you are coming through an inference to make that decision through the way in which you're interpreting those results in that clinical study. It's very similar from a computational study perspective. I would say arguably the bones of this are exactly the same. The details are very different, but when we look at a computational study, we have a number of aspects that we're choosing. So I've got a long list of things. You'll see examples of this later. You potentially have already seen plenty of examples of this, but we have to choose a number of things that go into the model. So that's part of it. We have to choose what we're measuring, or in this case, of course, computing. Things like stresses in the device, or the way the tissue is going to deform, or the way the blood's going to flow, all these sorts of things that we are choosing to observe in the model and the results from the model. But just like a clinical study, there are things that we don't necessarily know or have a direct insight into as it relates to, for instance, the comparison of the results of the model to a biological mechanism of action that might be not built into the model, or how a person is going to physiologically adapt to a given treatment. So there's aspects we don't know. There's a lot of things that we chose and we added into the model intentionally, and there's things that we compute or we measure. As with the clinical study, all of these things are wrapped up together in the model to give us an inference that allows us to make this decision from those observations of those measured quantities. So why should we trust the model, right? There's a lot of things that go into this, of course. There's a number of different boxes that have now shown up on the screen. It's a little bit more complicated. But as we look to derive trust in the model and demonstrate the credibility of that model, there's really two main things that drive that trust. On the right-hand side of the screen, we have the sources of the inputs and the comparators. So we are able, in constructing a model, of course, to leverage all of the information that's available to us in how we construct and make our decisions with respect to the construction of the model. Things like experimental data, scientific literature that might be performed by others, other data that's out there, clinical measurements, et cetera. So we have all this input information that's coming, and that we're able, not just input, but also comparison of the model predictions into that data. On the left-hand side here, we also have a whole list of credibility activities that we can go through. These are activities that somebody who's constructing the model has ideally done and is going to then ideally demonstrate to you from the assessor point of view. So these activities include the features that we have here, and the V&V section that's next will go into this in more detail. But fundamentally, the idea in the bottom highlight there, the rigor of these activities that one goes through is going to scale with the risk associated with the model in a very, very important way that Martha's going to go into next after I kick it over to her. But before we get there, let's talk about some of these concepts. So we can trust the model because of what we put into it and the activities we do. And we do that through these three considerations. These are very important definitions, and arguably they're going to be slightly different definitions from the way that these terms are traditionally used in other spaces. But when we think about verification, it is the idea that the computational model is going to accurately represent the underlying mathematics that are built into the model. So when we build a mechanistic model in particular, we have to make decisions about the mathematics underneath that model, right? Then we have to make sure that the computer implementation of that is accurate. So that's what we mean in our community by verification. Are we accurately representing that mathematical model? From a validation perspective, now we're starting to ask the question of, does this model accurately represent the real world? Validation is very, very important, of course. All of these are important. But when we look at validation, again, we're comparing the model predictions to the real world. And one thing that'll hopefully become clear, if it's not already, is that this validation can occur at different scales. So I used the word comparator on the previous slide. When we validate, we can make a prediction from a model, and then we can compare it to an observation in the real world. That might be an experimental data piece. That might be a clinical study. That might be a number of different things, but we're comparing the prediction of the model into the real world. So that's what we mean by validation. And then when we think about applicability, this is the idea that the validation activities that we go through should be explicitly relevant to support the context of use. Now, recall the context of use, the term I defined earlier, is this idea that you use the model for a very specific reason or for a very specific purpose in answering your question. The applicability says we've done some validation. Does that validation apply to the way that I want to use the model? Or is the validation in a completely different direction? Ideally, it applies directly. So those are three very, very important terms. Now, we've shown a lot of different things here, both conceptually and explicitly, with respect to how we think about modeling and simulation. The last thing I want to leave here as a tee-up for Martha in the next section on Beam V40 is the idea that we have guiding documents for the way that we do this. These are not just conceptual terms. These are put into frameworks that we can then work through to structure the way that we perform our credibility activities. Beam V40 is the keystone document in this space, and it provides this structured approach. Importantly, though, it is not a recipe book. It doesn't tell you exactly how to do things as in other standards. It gives you a mechanism for applying a framework. But this ultimately aligns the credibility activities that one is going to perform with the relevant question of interest that you're trying to answer. It does so in a very structured way, an important way that allows assessors and other people who are trying to interpret a model to understand what was done and how that model should be trusted or not.
So we will talk about establishing trust in silico generated evidence. So how started the story here? So the FDA and the industry were both interested in increased adoption of in silico data in regulatory decision making. There were significant concerns about using model-generated data since there were no standards around. But in 2011, FDA, the industry, and academia established through the American Society of Mechanical Engineers the Standards Verification and Validation and Certainty Quantification 40, a subcommittee to work on that. And they addressed the gap that they found by in 2018, the standard was published. And the name was Assessing Credibility of Computational Modeling through Verification and Validation and Applications to Medical Devices. So what the standards does provide a framework for assessing the relevance of adequacy of completed activities that established the credibility of a computational model. What doesn't do is to describe the specific validation and verification activities and rigor required to establish credibility for a particular device. So let's go through this using a practical and typical in silico evidence that many regulator or regulatory reviewers have seen before, and that is the MRI safety labeling. The labeling allows the clinician to know the safe limits. The most challenging labeling parameter is determining safe scanning parameters that doesn't create hazard for patients during radiofrequency heating. Let's use the family of spinal plate screw system as an example. That requires an MRI safety labeling. The central principle of the VMV40 is that the rigor of verification and validation activities should be commensurate with the risk associated with using the model for a specific decision. In other words, a model that substantially influences a high-consequence regulatory or clinical decision must be supported by stronger evidence of credibility than a model used for exploratory research, for example. Rather than prescribing specific tests of acceptance criteria, the standard provides a risk-informed framework to evaluate whether the completed VMV activities are sufficient for the intended use of the model. So the standard of framework introduces a structured process consisting of the following key elements. First is the question of interest, which describes, as said Steve, describes the specific question, decision of concern that is being addressed. The model must address a clearly defined decision of regulatory question, such as demonstrating device safety, predicting clinical performance, replacing a bench test, supporting a design optimization, for example, or evaluating labeling claims. The intended regulatory or engineering decision drives all subsequent credibility requirements. Here is the question of interest that simulation will address in the example. What are the safe MRI scanning conditions for a patient implanted with a spinal plate screw system given the risk of RF or radiofrequency-induced tissue heating? So let's go to a definition of context of use. As was explained before, the context of use defines the specific role and scope of the computational model used to inform the decision, and thus the model generate data has the primary or supportive role in the totality of presented evidence. The context of use asks how the model will be used, what aspect of the device performance it represents, whether it supplements traditional evidence, the specific decision being supported. The same computational model may require different levels of credibility depending on its context of use. In this case, in the example of the MRI, the model will be used to identify the configuration that creates the highest temperature rise in the standard of the phantom. In this case, the results from the in-silico model will be the primary use in that case, or the primary source of the evidence that is documented in the regulatory submission. A major contribution of the VMV40 is the concept of model risk framework, determined by two factors. So the model risk is the possibility that the use of the model may lead to the false or incorrect conclusion about the device performance, resulting in an adverse events for the patients. So, and all that we care about are the patients. So we have, in the case, the definition of model influence that is defined as a contribution of the computational model to the decision relative to the other available evidences like in vivo, in vitro, mathematical models. The icons here show the various forms of evidence, of course. In other words, the model influence indicates how much does the decision depend on the model versus other evidence. In the examples we can give as a low influence, the model is supportive evidence only. In a high influence model, the model substitutes for bench of clinical data, and combining these factors determines the overall model risk, which then drives the required credibility activities. As higher model risk, we have a stronger credibility evidence required. The decision consequence is the significance of an adverse outcome resulting from an incorrect decision. That from even a patching a scratch with a Band-Aid, or a disability, or even the death of the patient. So the risk increase for the risk of the patient. The decision consequence ask what happens if the decision based on the model is wrong. Examples of low consequence is the design screening. Example of high consequence is a patient safety determination.
Model risk is one concept is combining the two into a single model risk assessment. And that is, so model risk isn't, in this case, an aggregate of a model influence and decision consequence. So here's the very beginning of establishing some sort of gradation. That how much evidence, what quality of evidence, what quantity of evidence we need. So the model influence, we can currently, as an example, you can see if you can see the output from the model is a low or minor factor in the decision, or a medium with a moderate factor in the decision, and a high would be a factor in the high, significant factor in decision. And decision consequence, we establish a gradation, and an incorrect decision could lead to a very low, let's just say, it could be a health hazard, but a nuance to a nuisance to the patient or the physician, very minor effect. Or it could be a medium, could be result in minor patient injury, need physician intervention, or have other moderate impacts. Or it could be high, result in severe patient injury or death, or have other significant impacts. So we put them all together. Now we have a map. And this map has, so in this case, again, it's a proposed map. It's not mandatory. It's a good way of representing the combined effect. So you will have an area of low, so it's low moderate influence to low decision consequence to a high. And so as you go towards the higher the upper right corner here, you will have a higher evidentiary rigor. So both the influence is high and the consequence is high, therefore the risk is higher. So just look at the example for for the plate. So it's what would be a typical map of this one. A decision consequence will have a low adverse effect. Again, a decision consequence is really called out by the FMEA, could be post-market. All that information, this is what's been typically seen with this decision consequence, and it's considered very low. And the model influence, is high because we intend not to use any other evidence. And again, this is fairly typical currently, a workflow for all of the MRI scanner for MRI labeling for for orthopedic devices. Again, a decision is made purely what's coming out of an in-silico model. So in this case, we have a low-high. We identify a model, total model risk as to be a low-high. So let's talk about the second stage. This is a very technical. This is a technical part of the framework that's typically performed by a subject matter expert. So again, there are three main concepts that is converted to some sort of activities, and Steve covered those. The first one is verification. Did you solve the underlying mathematical model? And for in this case of a virtual coil or a virtual MRI, did we solve the Maxwell equations? And then and you could go from the mathematical representation to a numerical representation. Did you do all of them correctly? So there are a whole series of exercises you do that to make sure that the verification you are answering the did you solve the to answer yes, I solved the underlying mathematical model correctly. The next, of course, is the that's what you intended, but does the model represents the reality of interest? In this case, say for MRI labeling, again, we did a whole bunch of tests on a virtual coil. We put some rods, we measured the temperature, we compared the data, the results from the simulation to an actual ASTM phantom, and yeah, we did comparison. We collected the whole data to do that. And once we believe we have validated the model, the next question is the model credible for the context of use now? Because of course you don't want to your intent to apply this model to a bigger domain to answer the questions, right? That's your context of use domain. And this is your validation domain, right? The validation domain is much smaller, but the context of use domain is where your claims are, is much wider. So in this case, again, if you look at the MRI example, we have a plate that we did some sort of validation for one or two samples in that domain, but then we're going to say the context of use, my context of use domain, where I'm going to apply to, is going to be thousands of configurations. There are multiple lengths of plates. There's screws of different length and diameter. So at the end of this, I want to claim, this is basically to say to show that your model is applicable, or as what Steve said, the inference space is there, right, for the whole domain to be to work. So this is a little bit more of a detail of certain activities that needs to be done for the verification. Is code verification, calculation verification, as whole underlying sub-activities. Those sub-activities are actually the one that you do and you go through the gradation. I won't skip the details, but these are all the activities you need to perform to show that your model is credible. So as you develop this, you establish for instance, again, the MRI labeling, we have a case of low medium, and that low medium, these are your goals. When you do go through the activities, these are your goals of activities you need to achieve. So this is like an initial goals. This is where you think you should be landing in your credibility gradations. But then you go through the exercise, you might find you meet some of them, you may not meet some of them, you meet some of them are going to be higher, some of them could be lower, and then you go through this justification. You go into the assessment, and the assessments you need to justify, regardless, justify for each one whether how do you know you reached and you achieved what you achieved is enough, is sufficient. And then, of course, once you're done with that, if everything is you've achieved what you need to achieve, then it's the documentations part. And the documentation part is the last stage, and it's a very important stage. I highly recommend reading the FDA guidance on assessing the credibility of computational model and simulation in medical device submissions. So because the VMB40 does not provide any guidance on reporting, so this really helps a kind of a unifies or harmonizes what a report should look like, and this is really helpful. So I would highly recommend to download the guidance, read the guidance, and this will really helps you walk through all the evidence as it's being presented by VMB40 to be able to assess the evidence being presented. So two work streams that I think we'll be more talking about that in the 2-day session in London later on in October. And I talked about the model influence. You know, it is stakeholder. The reason we have stakeholders to comment or to provide input is because the model influence is subjective. And that subjectivity needs to be mitigated through the input from all the stakeholders, from the regulatory, quality, R&D, external regulatory, medical. They all have a say whether the assumptions, the question of interest, context of use, and your risk assessment is aligned with what their expectations are. And the trust gen, the other one that's the other colleague, so Ali Sarami is working on the model influence framework, and trust gen is led by Arzu Zakarian, is the Gen AI as applying the VMB40 to generative models.
Okay. So thank you very much, everyone, so far, and also thank you for the chance to present a case study that we worked through on some of this during the recent CEiRSI project. And I just have listed some of the collaborators that we worked with on this. Our case study was looking at a CPAP mask, continuous positive air pressure, and looking at the protection against rebreathing of that as defined by an ISO standard, ISO 17510. On this project, it was coordinated out of ResMed, but was done in close collaboration with some participants from the University of Warwick, University of Nottingham, Draeger in Germany, as well as the Therapeutic Goods Administration in Australia, and along with contributions from the CEiRSI group and the MHRA along the way. So let's jump in. Hopefully you can all hear and see this video. I find this quite striking, but this is an example of obstructive sleep apnea. This person is asleep, they're trying to breathe, but it's not happening. There's an obstruction, they're putting the effort into breathe, you can see the negative pressure on the chest, and eventually he's been roused from deeper levels of sleep, and that's brought innervation back to his muscles, opened airways, and been able to get some breath again. And so essentially, let's start with a little bit of terminology. An apnea is a cessation of breathing for 10 seconds or greater, and that video is a really striking example of that. And that comes from during our sleep, we all lose our muscle tone, and in some people that can result in an airway collapse, and that can progress to complete obstruction, as we've seen. But of course, when that happens, you have a reduction in oxygen and an increase in carbon dioxide. Your body's aware of that, so it springs to action and rouses you, restores muscle tone and airway patency, and you can breathe again and back to sleep. And this can continue many, many times in a night. And so these events, or obstructive apnea events, they're then essentially aggregated over a period, and it's a count over an hour for an apnea event or a hypopnea event, which is a reduction in breathing. and graded based on the number that you have during an hour. So in order to treat this, a common therapy, not the only one, but common therapy is CPAP therapy. The original research for that was done by Colin Sullivan out of the University of Sydney in 1981. And essentially the idea is if we apply positive air pressure to the airway, and that provides a pneumatic splint, and then allowing you to breathe normally on top of that. And so essentially you're inflating the airway like a balloon and allowing breathing. So that requires that you supply pressure. And this is done using CPAP equipment. So this is some of the things that we make at my company, ResMed. And we can see there's a flow generator that generates the pressure and flow, and a tube that connects it. And then you have a mask, which is positively held onto the head with some headgear, because it's pressurized, it'll want to fly off otherwise. And they're in a range of different form factors. And we can see here in the bottom picture an example of what that might look like. So let's have a look at the clinical challenge. So I'll introduce the concept of dead space. So dead space is basically the volume of breath that does not participate in gas exchange. So if we look at our airway on the right-hand side, the airway can be classified into two portions, the conducting zone and the respiratory zone. And the conducting zone does what its name suggests. It provides a transport for the airway into the lungs and the alveoli, where the gas exchange and respiration processes happen. And along the way, it will do things like condition the air and filter it and so on. But if we consider the conducting portion, it's not involved in the gas exchange. And so that's essentially a dead space. So as you take a breath in, not all of that breath is going into your lungs and taking part in the gas exchange. And what we're showing here basically is the anatomical dead space. And that's roughly about 140 milliliters everyone varies, but roughly about 140 mil. And so at the end of exhalation, the dead space gas volume will have a high carbon dioxide reading, about 5% by volume. And then during inhalation, we can see here approximately the first third of that is stale air. So we're not getting all of that air that you rebreathe in, and then when breathing out, again. So when we go and put something on our face, like a CPAP mask, we've gone and increased that dead space. And so the result of that is there's a potential to rebreathe more carbon dioxide, and that needs to be managed. So that can result, of course, in an increase in blood carbon dioxide gases, and that's exhibited in your end-tidal carbon dioxide and increased carbon dioxide rebreathing. So this needs to be mitigated in the mask by the incorporation of a vent, so that we can both maintain pressure, so not have a whopping great big hole there, but something that will provide a controlled venting and allow us to exhaust carbon dioxide from the vent. So ISO 17510 prescribes a physical bench test to demonstrate this. And it's a fairly complex test rig, but it consists of a CPAP system. So we have our flow generator, a tube, and then a mask, which is attached to a simulated head. And then along with this, we'd have something that would monitor the carbon dioxide, and then that would be attached to a lung simulator, which has a carbon dioxide source, which simulates a metabolic rate and in the human. And along with that, there is a prescribed waveform, which is called a sinusoidal waveform. It's got a 500 mil tidal volume. So it's not quite the same as how we would breathe naturally, but this is the prescribed form in the bench test. And it's a fairly complex test to set up. It can be sensitive to environmental conditions. With it being complicated to set up, it's also comes some expense in the equipment that's involved. And as we're using this to test the medical devices, these masks that we're making, there can potentially be long lead times to have physical prototypes that are ready. And all of that adds to some of the practical limitations and requirements of using this. So, as part of this project, we've considered how we might go about this using a modeling approach and using simulation results as part of regulatory evidence. Along the way, we used two different modeling methods. There was a continuum-based computational fluid dynamics model, as well as a lump parameter system model, which is took into account more physiology and was very fast to solve. I haven't presented that in this presentation now. I'll just be looking at the computational fluid dynamics model. So the image that we have on screen at the moment shows the domain of one of our masks, which has been sort of intersected with a representative face. And we can see tubes which go off to the inlet and then another one to the outlet. And so along the way, we prescribed boundary conditions there. At the lung, so they were set to atmospheric pressure at 0 at a remote boundary with trace levels of carbon dioxide. And then this is an example of the computational domain that we used, and it was graded to capture the geometric complexity. So we had, for instance, enough elements to refine a velocity profile through our vent holes, but hopefully find as well a balance between what we could reasonably solve in. And one of the great benefits of CFD is it provides not just numbers, but it provides insight and understanding. So looking at the results that we have here, we can see the concentration of carbon dioxide, which is shown in the animation that's on loop play at the moment. And we can see that the concentration is, during exhalation, you can see the carbon dioxide rich air coming down the tube, and then that is delivered into the plenum of the mask where it is mixed, and we can see that some of that is vented out of the atmosphere, out of the vent into the atmosphere and removed from the system that the patient will breathe. So we can just visually see that we're removing carbon dioxide. And if we have a look here, we can look a bit more quantitatively. And so we can track our gas levels past a sensor location, which would be commensurate to the test rig. And we can see that this has converged over a few breaths, and we can see how and when things are working. So we gain understanding. We can also do this early in our development cycle. We don't need to wait for expensive prototypes, and we can scale it. So once we have these models prepared, if we're smart about how we do it, we can prepare and run multiple configurations simultaneously. So following on from what Martha presented, we had a specific question of interest. So we followed the VNV40 framework, and our question of interest is basically defined under ISO 17510 by the limits that we have there of not exceeding normal end-tidal CO2 without a mask by more than 20%. And so we're able to measure this from our model. And then we defined a context of use here. So in this case, we're proposing this to be a surrogate for bench testing, in which case our model influence becomes very high. But we do have bench testing available for validation. So when we look at model risk, so as I just mentioned, our model influence is very high. And so in this case, we called it controlling. So we're quite toward the right on the horizontal axis on our model risk graph. And the decision consequence, where we landed on our risk review, was around moderate. And we considered the consequences of making wrong decisions and the likelihood of adverse outcomes from that. And so working along this framework, we then used this, we landed at this sort of high level of model risk, and we use that to inform what level of credibility assessment we use as we work through model factors. I won't go through all of these, but we went through the verification, validation, and applicability, but essentially the little color scales, blue on the left and green on the right, were for our CFD and systems model respectively, and essentially think of them as a percent of rigor that we proposed for each of the credibility factors, and so we would work through those, and some would be higher than others, and some would be done inherently as part of doing the work, and some would require specific effort. And then now looking at the benefits and impact of this, the CFD, and we can also look at comparing here to our bench test, and we can see that we landed quite close. We did one particular run because it took too long for the life that we had in the pilot project here, but we did one particular run and we were close enough. We were well within margins of our 20% limit. And we really like that we could use different modeling techniques, and that allows us to look at the same problem in different ways and increases credibility. The CFD provided insight into the performance and was able to visualize our flow field and mixing, and we can use this to guide our design.
So I'm going to very briefly try and cover the neurovascular. We looked at a variety of things in some of our pilot studies as part of the UK CEiRSI work that we did about just over 12 to 18 months ago. And brain aneurysms are one of the relatively common things that we see worldwide. Essentially, they're a weakness, a bulge on a blood vessel in the brain. They can rupture, cause subarachnoid hemorrhage, and they can cause enormous levels of death and disability, resulting here in sort of 50% of some sort of deficit in survivors of subarachnoid hemorrhage. And treating them historically has been surgical. So you operate, you open the head, you put a clip across the neck of the weakness, and that obviously carries its own risks in terms of surgery and retraction and things like that. So the treatment's been evolving over the last 30 years, initially with putting little platinum coils inside the aneurysm to close occlusion. And more recently, what we've seen is treatment with flow diversion. And in flow diversion, what you have is this braided stent that changes the flow through the artery so that you reduce the flow inside the aneurysm. And what happens then is you get stagnant flow and eventually clotting, and eventually you'll get endothelialization of the vessel, and so exclude the aneurysm from the circulation, which obviously prevents further hemorrhage. And that kind of has been the way things have been developing probably for the last 15 years. We're seeing more and more aneurysms treated by flow diversion. And what you measure really is this immediate reduction in flow in the aneurysm during your angiogram sequence. You can't really tell exactly what's happening. It's a surrogate marker that we rely on, and the patients require imaging, and typically measurements are considered successful at about 12 months. Let me just go on. So the first flow diverters are probably approved by the FDA in about 15 or 16 years ago, about 2010, 2011. They're limited at that stage to these large, wide-necked aneurysms that are not felt to be suitable for coiling because the coils might prolapse out and occlude the vessel. So that was the sort of the concern, and that's why there was a bit of reluctance to approve these things more widely. And initially, the FDA approval, at least, was really quite restricted to a small section of the internal carotid artery and a specific morphology of these wide-necked aneurysms. Small aneurysms or medium aneurysms weren't really included. So obviously, as things do with surgery, people said, well, why can't we treat smaller aneurysms or different sized aneurysms? So there were more trials over the years, And several years later, you see this trial with about 140 odd patients. And the FDA then agreed to expand the indication to these smaller and medium-sized necked aneurysms. And the question really for us was, could part of this evidence have been generated using some sort of credible in silico trial? So could this computational evidence have supported this indication expansion and maybe reduced the size and cost or even duration of the clinical study? So what did we do? Well, essentially what happens first is that we start generating virtual patients. So we generate anatomy, we generate physiology from measurements. We then create this virtual population for our trial, and each point is a virtual patient. We then simulate the deployment of our stent inside each anatomy. Now, I would say at this point that we assumed the stent placement was good. We did not look at challenges of stents where they're not properly placed. And again, that's common with any surgical trial, that the assumption would be that, the stent is placed correctly, the procedure is followed. So there's nothing particularly unusual about that. But we then use computational fluid dynamics to actually predict the blood flow after treatment and actually quantify that reduction in flow inside the aneurysm. And depending on how complex you make the models, you can also predict, thrombus formation, other treatment outcomes. And you can then develop this virtual clinical study that can kind of complement your traditional clinical evidence. And so what does the model add? Well, here we're not aiming to reduce or replace a clinical trial, but we're wanting to provide additional evidence for an indication for an already approved device. We know a lot about these devices. They've been implanted in lots of patients. We know what the risks are. We understand the surgical procedure. We understand the deployment risks. So here we understand a lot of the clinical risks. What we're really looking at is, given that we know those things already, could we reduce the number of patients? And could we shorten that development timeline so that these devices that we already know are pretty safe, certainly from a clinical perspective, from a neurosurgeon's perspective, we know these devices are safe. So could we get into patients earlier? And ultimately, could we reduce this evidentiary burden, but also maintain confidence in the regulatory decision? So the real question here is not whether or not the model is absolutely perfect, because as you've heard from Sam and other people, no model is perfect. They always have limitations. And clinical trials by themselves, as Alex is fond of saying, are models, and they have very specific entry and exit criteria and very sort of tight characteristics that allow what we're predicting. And they don't predict everything because they can't. And so the question really is this model credible for its intended use? So if you follow the VMV40 framework, the question of interest here is, could a flow diverter achieve a 12-month occlusion rate in small aneurysms compared with, the currently approved indications? And the computational model was used to predict that reduction in blood flow immediately after the treatment. And that was our surrogate for long-term aneurysm occlusion. And we know that those two correlate, and specifically clinically. So we evaluated, first and foremost, what the model risk was. And the main clinical harm here is incomplete occlusion. So the aneurysm isn't completely occluded and isolated from the circulation. That happens in about 15% of patients, typically, in a clinical study. It's not terribly unusual. It's manageable. There are things you can do about it, and it doesn't need an urgent surgical intervention or anything like that. So we came to the conclusion that the decision consequence here is medium. And if we looked at the model influence, because there was already substantial clinical evidence, and about 30% of aneurysms in the previous studies are small, the regulatory decision here is not going to rely solely on the computational model. So we came to the decision that the model influence was also medium. And so overall, we think the overall model risk here is medium, which then determines, as we've heard, what level of credibility evidence is required. So we're going to look really at clinical evidence or clinical validation today, and benchtop is important, but I'm not going to sort of cover that today just because of time. There we go. So if we consider how to clinically validate and use this FD-PASS exemplar in-silico trial. So we wanted to determine whether or not the in-silico trial could actually reproduce the results of the pivotal clinical studies. And so we applied the same inclusion exclusion criteria as the original studies. And I think if you just see here, so this is what we end up with. We can also model some other things. We can look at normotensive patients and hypertensive patients, and you can see there's a difference, which is perhaps not surprising, given the physiological changes that occur in those conditions. And our FD-PASS is able to, match these clinical results very, very closely. Which is really encouraging, and it gives confidence that the model captures the relevant clinical behavior and that it can provide this meaningful evidence. So just to kind of sum up, so I'm back on schedule time-wise, this case study does seem to provide credible clinical evidence, credible evidence that in-silico trials can complement the clinical evidence for this indication expansion. And there is, I think, probably good evidence here to suggest that they have the potential to reduce development time. They can lower the evidentiary burden. And what I mean by that is not to make it a lower bar, but to reduce perhaps the numbers of patients, the time taken, and therefore get these devices to patients who, particularly in this situation, we know these devices are already routinely used. We understand their safety profile. We're not talking about a brand new device and a brand new indication. And I think that if you support your in-silico trial with appropriate credibility assessment, the computational evidence, it can become reliable, and it can become a sort of really reliable component of that regulatory decision.
So I'm also going to present one of the UK CEiRSI ELOC projects that we had, and that was about transcatheter heart valves. There was the pilot 4. This is the panel that we worked with this time. The clinical challenge to aortic valve pathologies is here illustrated. I mean, you can see on the left-hand side a healthy aortic valve, an open and closed manner. Then you can see on the right-hand side the diseased aortic valve. I mean, this structure should show that there is some type of sclerosation and also calcification of the valve. So the valve is actually in systole not opening appropriately and in diastole not closing appropriately. That's the issue that we are facing. In the treatment, we have different treatment options. Back in the days, of course, we tried to manage blood pressure because they're one of the things that won't, secure the heart muscle. But I mean, ultimately, we have to do something. If the valve is damaged, we have to replace it. Gold standard for many, many years was actually cutting off the thorax and doing open heart surgery, replacing the valve in a surgical manner. But since the patient population is evolving, we're getting much more older patients, more patients that are quite sicker, that can't withstand all the side effects of an open heart surgery. On this basis, and also like with the developing transcatheter techniques, the so-called balloon valvuloplasty was developed many years ago also already for ultimate treatment of patients that can't receive any surgery. So the valve was just pushed into the aortic annulus to just rupture the calcium, then leave the valve in place. And usually the valve opened a little bit better, closed not really better. and that had a lot of issues with so that wasn't really helpful, and it had also to be repeated within a couple of months all the time, because it started getting together. And this is when transcatheter heart valve replacement was developed, where we actually have a stent frame, and in the stent frame is a biological valve. And this is going to be put in place and stays in place. It's a wonderful technique on one hand. However, we still have unsolved issues. That was actually the clinical challenge that we took on for this pilot phase. We have two issues with transcatheter heart valve implantations. That's the risk of pacemaker dependency after transcatheter implantation of the valve. And it depends on what type of publication you look at, but I mean, it ranges between 36 and 47.5%. That means after the implantation of the valve, the conduction system of the heart is going to be impaired. That means that the heart can't beat fast enough. So these patients may then also require permanent pacemaker implantation, right? Another surgical procedure. So first you get the valve, then you need a pacemaker. And the second aspect of transcatheter valve replacement that we are facing is actually the paravalvular leakage. Here you can see like schematically, the valve is put into the annulus, and then if you have a paravalvular leak based on calcifications that will change this round shape of the aortic annulus, so that the blood is actually not passing only through the middle of the valve where it should go, but also passing through the sides of the valve. This is, on the long run, constantly putting pressure and force on the ventricle that leads actually to a higher mortality during the follow-up. So these two factors are still present when we do transcatheter aortic valve implantation. But however, it's also correlated with each other. So if we implant the valve, if the implant pressure is too low, we will generate paravalvular leakages, right? So then the blood will go through the sides of the annulus. But the other thing is, if the implantation pressure is too high, then we are going to impair the left bundle branch that goes, or that actually passes through the annulus, and then we have the risk of pacemaker dependency. So that means there are some devices that has lower risks, some devices have higher risks. However, we have to understand this correlation. And this is a limitation of traditional evidence. So we do not know exactly the influence of this pattern mechanism. So how much is actually the challenge based on the calcifications? Or is it the position of the valve leveling, lower or higher in the annulus, and also the pressure applied? So with these traditional data, we as physicians cannot really provide a prediction to the individual patients about paravalvular leakage and also pacemaker dependency. And of course, the only thing that we can do, we have a high dependency on clinical investigations. And we all know that this is, of course, also a model, because we have inclusion-exclusion criteria. Patients are highly selected. This is where this modeling approach came in. So we said, okay, we have here 3 harms that TAVI can cause. And this actually translated into our context of uses. So that means first context of use, we couldn't even only have one context of use. So we had to decide for 3. So one is actually the position of the valve in the annulus and the hemodynamic function. The second is the position of the valve and the impact of the conduction system. And the third context of use was the position of the valve and then paravalvular leakage. The question that we had based on the modeling approach, when we would like to change a part of the valve, of the stent, the design of the stent, could this be then, first of all, reduce the risk for pacemaker dependency, for paravalvular leakage? And would this be possible to get an approval with a reduced number of patients leveraging In-Silico evidence? That was our question of interest. Translated, that means we will minimize risk of paravalvular leakage and we will have minimal impairment of the conduction system. What a model adds. In the green area here, you can see predicate device. We have a lot of bench testing based on 5840. We have the clinical trial that was done. And of course, with all these data, we got an approval for a redesigned device. You should, traditionally, you should also have to do all of this. However, if you don't know what actually you need to change in order to change the incident of paravalvular leakage and conduction disturbance, you could run in-silico clinical testing first with a redesigned device. You could run an in-silico clinical trial, and if this would provide some evidence saying, okay, we can actually reduce the risks, then we could combine this evidence to get an approval with in-silico evidence. What does this add for manufacturers? I mean, definitely add something with regard to the device design, also with regard to iterations. What is for this model with the physicians? Actually a prediction of an optimal position of the valve, optimal implantation pressure, and of course also an optimized communication with the patient, also with regard to the risk of paravalvular leakage and pacemaker dependency. The model risk here was rated medium-high for contact of use 1, the device component, medium-high for conduction disturbance, and for paravalvular leakage, medium. For regulatory use and the evidence level, what we could add with this model, we could add an in-silico trial with the redesigned device, in-silico device testing with the redesigned device, and a clinical trial with a reduced sample size. What we get out of this, we could evaluate the design change before actually building a new valve, also using, of course, the standard, that is the 5840 standard. We would have validated, transparent, and reproducible outcome parameter, and an estimation of risk. And in the end, of course, an understanding of these unknown pathways that you impossibly can't measure in vivo. One other important aspect here is what we always have to face, that even if we have clinical trials on predicate devices, there might be an over-reliance on predicate data. So who can tell us, and with all these highly selected patients, if this is the only truth? The same with evolving populations. So we are not having this huge amount of patients that we can enroll in clinical trials anymore. And of course, we have to think about, even if we are refining, right, where to put the valve. I mean, this procedural learning curves and then device refinements. I mean, and this is something that would, coming out of risk from traditional clinical trials, that could also be leveraged with in-silico data. Benefits and impacts for this type of study would be a lower patient number in the clinical trial, faster development without multiple iterations. We will increase safety profile of the device by amending the device design. So safety by design, actually, because we also tested it against state-of-the-art, the data that we already have, and increased predictability for possible adverse events. And this also might lead to that physicians actually consider treatment strategies based on more patient-specific data. And last but not least, an increased level of optimal patient management and public health. We shouldn't forget, I mean, modeling of data before design changes will reduce definitely the time to market, and also the cost that will come for public health if you have to implant the valve first, and after that also multiple treatments for pacemaker dependency, so adding a new implant or also treating paravalvular leakage.
Yeah. So while this webinar is mostly focused on medical devices, I want to highlight that most principles introduced can also be applied to pharmaceutical products following the ICH M15 guidance on model-informed drug development. And we are also aware that there are several people in the audience who are particularly interested in pharmaceutical products. So we added a very short case study on pediatric physiologically based pharmacokinetic study with material very kindly provided by Dr. Mary Malamatari from the MHRA in the UK. And so we hope that this will give you a little bit taste of what in-silico for pharma will look like. And I also want to highlight that there will be more case studies specifically focused on pharmaceutical products in our future events, including our in-person London event in October this year. So basically, this study concerns AutoWork, which is a protein that temporarily replaces the missing factor VIII needed for effective hemostasis for patients with severe hemophilia A. And the regulatory interest here is to find the reliable dosing recommendations of AutoWork for children where direct PK data was very limited. And in particular, there were no clinical data in children under 2 years old. So basically, the team developed a PBPK model to simulate AutoWork with age-dependent physiology incorporated in the model, and then they evaluate the model using the available relevant clinical data, which are Eloctate, which is a similar drug in adults and children, and for AutoWork, which is only available in adults. And after validating the model, the team generated in-silico evidence to simulate the response of AutoWork in children at 3 different age groups, which are from 0 to 2, 2 to 6, and 6 to 12 years old. And the predictions were compared with observed PK data wherever possible. So when we look at the regulatory interpretation and decision using this in-silico evidence generated, we need to follow the newly developed ICH M15 guidance, which adopts a very similar model risk informed framework as the ASME 40 framework that has just introduced. So basically, the model credibility requirement for based on ICH M15 depends on the model risk, which consists of the model's influence on decision and the consequence of an incorrect decision. So here you can see a very similar matrix as we see in ASME 40 in this paper led by Kaiwash and co-authors. And back to this pharmaceutical study, we evaluate the model following this framework, and you can see here that the response of AutoWork were validated in adults, which is the data available, and the response of Eloctate, which is a similar drug, was also validated in pediatric participants from 2 to 6 years old. But like we have mentioned, that the patients younger than 2 years old is not available. So with this framework and the evidence generated, we can draw the regulatory decision that the PBPK model is acceptable to support dosing in children older than 2 years old. However, when applying the model to replace clinical studies in pediatric population younger than 2 years old, which is considered a high regulatory impact case because validation for this patient group was not performed in the study. So overall, the take-home message is that the PBPK models can bridge pediatric evidence gaps, but regulatory acceptance also depends on model credibility. And following the risk-informed approach in ICH M15, the model credibility is also established through verification, validation, and uncertainty quantification, and which should be commensurate with the model risk for a specific context of use. And finally, I want to acknowledge great support and help from MHR colleagues, including Mary Malamatari, Susan Cole, Andrew Butler, and SM Kaiwash.
Just to very briefly bring things to a close. So hopefully we made the point that there is a number of changes that are happening in the way that we are doing medical product innovation, and we focus today primarily on devices with, but we want to make sure that it is clear that similar techniques can be used in the pharmaceutical domain. We spoke about the ASME BMB40. There is also the ICH M15, which is kind of the equivalent adaptation of ASME BMB40 to the pharmaceutical domain. And we will have separate sessions in the future for, dedicated to pharma. We felt that it was too much already to sort of try to mix all the elements. But broadly speaking, in both devices and pharma, there is a need for better tools or complementary tools. We believe, and hopefully we have made some steps towards conveying the importance of CMS in the future, and also that where do you start? Because this is a massive change in the way we think in some ways. And what we have seen in conversation with a number of colleagues is that focus primarily on the use of in-silico there where there are evidence gaps currently in the regulatory process, And we provided one example in the area of pediatrics, but you also have rare conditions, combination devices, areas where perhaps there isn't an alternative to in silico methods, and where at least this can provide some clarity to what is avoiding evidence. Some of you have already expressed in the pre-webinar survey that this is not something of the future, that is something that is already happening, and we believe so. But also we believe that it's really important both to catalyze that uptake, to have both pull from regulators and push from industry. And hopefully you've seen that in the collaborative work that we've done through the UK CEiRSI, effectively, that's what we try to build, a co-creation by bringing all the parties along. And you've probably also seen that while this initiative has been funded by the UK government, it has been open and remains open to participation across the globe. So we don't want it to be exclusively UK focused, because we believe that harmonization is critical here as well. Something we haven't spent time in this webinar, but we want to reassure you that is very central and important to us, is the working alongside patients. And we have had a number of public and patient engagement in sessions where we have taken some of their input and advice as well. And for instance, just to give one piece of advice that came from them is the usefulness as well of in silico methods as a way to understand the risk for them to participate in human trials as well. Okay. Something that is also coming, in many discussions, is that there isn't really a gap of legality in many cases, but a gap in confidence. So the need, and that's why events like this that trying to share our understanding and also hearing, hopefully, your feedback are so important, so that we create sort of authoritative, positive frameworks that sort of turn, as we put here, tacit allowance into trusted adoption. Okay. I remind you that we will have a more deep dive event in October this year. It's going to be the first edition of what we hope will be a sort of an itinerant training program. So if you, in your geography, you are particularly interested to host a subsequent version of this event, to bring it also closer to, your colleagues, drop us a line to Mark and myself, because we are very keen to, as we develop these editions, being able to sort of bring them to definitely to Asia and to Americas as well. And then questions that remain to be answered. So we appreciate a lot for the 55 plus people that provided answer to our pre-webinar survey. And we identified a number of areas that we will look at reinforcing in the upcoming training program. So your input has been really heard. And just also to get you an idea, this is the people who contributed to the responses at the time we did the snapshot. But we have had quite a lot of representation from both regulators and approve and notify bodies. If you haven't answered the survey, let us know, because we hope that there is still an opportunity for you to complete that. And the more information we have that we will share back with the agencies will help us to understand how we can take things forward. And with that, I just want to say a big thank you to our faculty for all the time, engagement and expertise they brought. They've done this, voluntarily and out of, their regular duties. So this is really, really appreciated. Also to all of you, the 80 plus people that contributed to attended the event, and finally to IMDRF for the opportunity to share this knowledge and hopefully learn as well from members. And if there are any questions or if you want more information, we leave you there a couple of contacts to probe further. And with that, just to say a big thanks as well to Michael and Lara, who helped with all the logistics, and Roberta as well from Avicenna Alliance, and look forward to continue the conversation with you. Have a great rest of the day.